Their ecology lab. I just want to make a video here to guide you through some of the stuff that you'll do in lab number 8, where we're talking about null models. The data set you're going to be working with comes from a very, very famous study from back in the 1940s or 50s by this guy, Robert Macarthur. And Macarthur was working on the coast of Maine, and he was really interested in these 5 species of birds, these 5 warbler species, wood warblers. Now, what's interesting is that these birds are really similar, right? They're similar size, they have similar behavior, they eat pretty much the same thing, they mostly forage for insects, largely mosquitoes. They occupy the same habitat. They're mostly foraging in balsam fir trees. They nest in the same kinds of places and so on. And Macarthur had this question about how do all these species coexist if they seem to be occupying the same niche, right? Remember, we talked about niches, and that's sort of the role that a species plays in its community, the resource requirements, their habitat, their diet, and so on. These species all seem to be occupying the same niche. Why doesn't one outcompete the others? How can they all coexist if they're so similar? So to answer this question, we need to do a couple of things. Number one, we have to decide how should the niche be measured. And there's lots of different ways that you can measure and compare niches because any of the resources that a species needs is related to the niche they occupy. But some studies have shown that the most sort of significant partitioning, the way that you can separate species in niche space, is by looking at microhabitat, so very fine-scale habitat characteristics, and by diet. Okay, then we need a way to measure or to quantify niche space. And so with this data set, we're really looking at overlap, right? How much time or how much, to what degree do the different species use the same resources? And so in this study, Macarthur was looking at how these species forage, how these birds forage in the canopy of these different trees. So if they're foraging in the same location within the canopy, are they, how much time are they spending in the same spots versus how much time are they spending in other spots? One way to quantify this, there's several different ways, but one way is Piankas Index. And that's the equation's shown here, but it's basically you're summing and looking at the amount of time species 1 and species 2 spend in the same place or using the same resource. So Piankas Index ranges from 0 to 1. So 0 would mean the 2 species do not overlap at all. They don't spend any time in the same location. And a Pianka value of 1 would mean they spend all their time in the same location. So your values can vary. Okay, so Macarthur, what he did was he spent thousands and thousands of hours observing these different species as they're foraging in balsam fir, and he has these little schematics of the tree canopy, from 1 at the top to 6 down below the canopy, and then from sort of the basal area inside near the trunk out towards the terminal branches. And so he divided into several zones and then made careful observations of how much time each species spends in each zone foraging. And when he compared these graphs, he saw that, well, the different species are spending different amounts of time. They're specializing in different parts of the canopy. So he proposed that, well, what these species are doing and how they manage to coexist is that they're partitioning their niche. So some of them are specializing in lower parts of the canopy and some in other parts. And so this became a really, really famous and influential paper and really famous sort of line of thinking about species coexistence and biodiversity. But one problem that ecologists realized later is that, well, species would be expected to overlap anyway, even if competition isn't driving them to partition their niche. So we need a null expectation here. We need a null hypothesis, and that would be how much niche overlap would be expected in the absence of competition. So if competition isn't important, we would still expect some overlap. So what would that be? And that was articulated by Connell here in 1980. It's not enough to show that species differ in their use of resources in a way that reduces niche overlap. Even in the absence of competition, species will differ in their resources. So to answer this, we need a null model, and that's what we're going to use. These are the results from EcoSim. EcoSim we've used before for rarefaction, and it's a null model because it basically simulates assemblages of species and how they occupy their niches. So what we see here on the left is this is a distribution of 1,000 simulations of those warblers. So we take those 5 species of warblers, and we simulate. We're throwing the birds onto the tree, seeing which niches they occupy, and then doing it 1,000 times over and seeing this distribution. And then you can see the dashed lines here are our 95% confidence limits. The red line up here at the top, that's the observed values. That's the values that Macarthur directly observed. So we can calculate a Piankas index for Macarthur's value. And over here on the right, we can see the observed index is 0.55514. That's the Piankas index for his observations. Now, when we simulate all of these and we get this distribution, we see the mean of the simulated index of those 1,000 simulations. The Piankas index is 0.39. So that's lower, and it's quite a bit lower because you see all these simulations, the observed value falls way above the 95% confidence limit. So the observed is really different than just simulated data, and the simulations don't assume any competition. We're just throwing birds on the trees. So what this shows is that the observed data, Macarthur's data, actually has a higher Piankas value, more overlap than we just expect due to chance alone. Now, if Macarthur was right, he would, and competition was driving species to partition their niches and overlap less, his data should be down on the low tail. It should be less overlap than the simulations, but his is wrong in exactly the wrong way. So we're actually seeing more overlap than you expect due to chance alone. So this is really interesting because this sort of fundamental foundational paper about coexistence, when you actually put a null hypothesis in there, it's wrong. So what you're going to do in this lab is you're going to load in that tutorial data set into R. And so you should use RStudio. And what you'll do is you'll download this from the website. The instructions are in the lab manual. And you'll have this set of files in your folder. And you're going to load in this one, which is right here, EcoSim R niche overlap shell. And that's what I've got up here in my upper left window. And the data are also in here. Here's Macarthur's warblers as a comma-separated values, a .csv file. And so what you'll do is, all you have to do is run every line of code in this script. So up here, we clear all the data. Here we set the source. And then we set up the parameters of the model. So you can see we're using Piankas Index as our metric. There's several different model choices, and those are also explained in the lab. So randomization algorithm 3 is the default, but you can also choose 1, 2, or 4. And then, 1,000 repetitions and so on. When you run this, then down here you execute the analysis. When you run this, you'll get a figure just like I just showed you, where you'll see the distribution of simulated values and then the observed values there. The other thing to keep in mind is you'll run this multiple times, so you'll run it with different assumptions of how we simulate those data. And then you'll see if we can run this model in any way to sort of support Macarthur's hypothesis, or if he continues to be wrong. So work through the lab, and if you have any questions, I'm available by email.